Hello, my name is Truman Price. Today I'll be presenting to you on the wi38 cell line for MCB 150 discussion number 10. To begin, I'm going to give some general knowledge on mitosis and cell division. Uh, Cell division occurs when a parent cell divides into two or more daughter cells. Mitosis is one of the cell division types in which the parent cell creates two identical daughter cells, usually done in somatic cells. So that's everything except germline. So you think like your heart cells, skin cells, anything not sperm or egg. It consists of 5 basic phases: prophase, prometaphase, anaphase, telophase. And this is during the M phase of the cell cycle. And you can see in this picture all the different generalizations of it. All right. For prophase and prometaphase, beginning with prophase, chromosomes will be condensing into visible chromosomes, as you can see here in the bottom right picture. Each chromosome consists of 2 identical chromatids connected by 1 centromere. So we can see for the X structure there's an X, but actually it's 2 chromatids connected by 1 centromere. This was done during the S phase, synthesis phase, where you replicate all the DNA, so you have 2 chromosomes. So we can see those will get split into 2 different cells. Nuclear envelope breaks down and spinofibers begin to form from the centromeres and eventually move to opposite poles of the cell. By prometaphase, usually these spindles will be on opposite sides and begin moving to the nuclear parts of the cell and attach to kinetochores, as you can see in this bottom middle picture right here. All right, for metaphase, pretty simple here. Spinofibers will attach to centromeres of the chromosomes, and chromosomes will align across the metaphase plate, which you can see is like this line. We can see how the 2 chromatids will be facing different sides of the cell, eventually splitting open. All right, for anaphase, spinofibers will shorten and cause the sister chromatids to separate and be pulled to different poles of the cell. We can see here the cleavage furrow is beginning to form, so we can see where the 2 cells are about to be formed, and it foreshadows how 2 identical daughter cells are formed. So you can see the chromatids are separating. We can see these are going to be identical because they're the same chromosomes. In telophase, the new nuclear envelope will form around the different sets of chromosomes. We can see these cells are beginning to detach through actin fibers. And chromosomes will begin to decondense so that genes can be expressed, and spinofibers will break down again. All right. For origin of the wi38 cell line, these cells were first taken from a lung of a female fetus which came from a legally aborted pregnancy. These lungs were packaged and delivered to Northwest Stockholm and then eventually the Wistar Institute in Pennsylvania, and there Hayflick would unpack it and place them in a flask of enzymes for maintenance and test on them. That is in 1962. Characteristics of the wi38 cell line: these are diploid human cells with the normal female karyotype, so 46XX, as you can see in the bottom right. You can see it in the slide. Sorry, my camera, I can't really move it. Sorry about that. I might try and move that later. Genetically stable, non-cancerous, and pathogen-free, which will be very important for later. These cells would double about 50 times before reaching senescence, which is the Hayflick limit. He established this, I think, before the wi38 cell line. In this 1961, which is a little before the serial cultivation of human diploid cell strains. That's his paper on the Hayflick limit. He found that even if you froze them, these cells would actually remember what division they were on, and it would reach about more so 50 divisions. This is because telomeres slowly decay after each replication. They'll shorten due to the end replication issue, and by the time we get to 50, they will be gone, so the cell knows not to divide anymore because might get into actual expressed DNA. We see in this graph on the x-axis days in culture, we can see in phase 1 doesn't replicate because there's accumulated population doublings. As we get to phase 2, it's pretty consistent. It's going at a constant rate, and eventually it'll slow down a lot by the time we reach 50 and stop, basically. All right, for maintenance of the wi38 cell line, This was grown in a culture medium, supplemented with fetal bovine serum and some other enzymes to keep it alive. And the bovine serum is enough to give it enough nutrients to survive and keep replicating. Antibiotics were used to lower bacterial contamination. It was incubated at 37 degrees Celsius. This is just optimal temperature for these cells, and frozen in liquid nitrogen if they wanted to do it for long-term storage. And we'll see that later. What are some of the medical uses of the wi38 cell line? The wi38 cell line had widespread uses in vaccine creation and finding cures to illnesses by providing a reliable environment for growing viruses. As I said before, it was genetically stable, non-cancerous, and pathogen-free. So vaccines for rubella, rabies, and chickenpox at their said dates more so are some diseases that now have vaccines due to the use of the wi38 cell line. And by the 1990s, it was actually being used to research cancers. You can see in the bottom right here, this little table. So I guess for my examples, I'll say rubella. We can see the vaccine annual cases it was getting, and then pre-vaccine annual deaths, and then cases that were averted with 95% coverage of the population. Deaths averted by 95% of the population. I'll tell you right now, it's at least in the millions, at least 100,000s, and deaths are probably in the thousands. So it's truly incredible. For each of these, how many lives this cell line was able to save because it was so well able to test these viruses on and create vaccines by growing these viruses and testing on them. So yeah, the cell line was it saved millions of lives just by being very useful for growing those viruses for researchers. Okay, here I gotta give you some just general history of the cell line, the science parts. Hayflick derived these cells from a 3-month-old aborted fetus as long, around 1962 when it got delivered. Named after the Wistar Institute, so wi38. It became mass-produced by splitting cultures onto new dishes after its useful and safe properties were discovered. This was with the help with Hayflick. He got Stanley Plotkin, supposed to say virologist. He confirmed that these were safe to test viruses on. And it was then used to grow viruses and create new vaccines. And yeah, I think that's about it. Yeah, yeah. So those two figured out this could be a very useful cell line. You can see here. I know this isn't lung tissue. Just pretends it could be this either. It's just fetus, the kidney tissue, or lung tissue, whatever you want. And then you take a human cell and you grow it, and then you can see you split it into even more cultures, and then you test and do whatever, make more, and yeah. Here's a general history of the cell line in terms of the legalities. There's a lot of legal issues and conflict going on. So Hayflick finds out his partner Kaprowski and the Wistar Institute, were basically profitizing and monetizing his cell cultures that Hayflick was originally just giving out. This happened all in 1968, and he felt injustice. Practically had no ownership of the cell line. He felt pressure to sign. I think there's one paper or one contract where he would get 10 of the remaining 370 ampoules, and yeah, he basically felt that was not fair since he was the one who discovered it, and he was basically losing ownership. So when he moves to Stanford for a job, and he actually steals all the ampoules remaining, brings them to Stanford, and starts selling them on his own at the same rate as the American type culture collection. I think that's what that is. They're like a nonprofit who normally would give out cultures. And the NIH publishes later that Hayflick sold U.S. property and causes Hayflick to go unemployed. This is 1976. Researchers then feared the loss of the cell line since there were so many legal issues going on. I know Hayflick then he finds a job at some children's hospital. I think eventually is protesting for his rights to have ownership of that cell line. In the summer of 1981, Department of Justice reached a settlement where Hayflick gained the original 6 ampoules and 90,000 in proceeds from originally sold cells. So, and I know he basically made no profit off his cell line in the end. So there's definitely a little bit of injustice here that he wasn't able to have full legality and control over something he discovered. But I'll give my thoughts on that later. What are some ethical considerations of this cell line? At the time, there were actually no ethical standards for the use of fetal tissue, and the mother of the original fetus did not give any form of consent that was known, and the abortion was done for medical reasons specifically without any knowledge on the possible testing of on the fetus's tissue. So is this an ethical violation against body autonomy? Arguments began, I think, around 1970. Some argue that since the research was life-saving, it's justified, and some argue that simple lack of consent is definitely concerning. It's a little strange that you have no idea that your fetus can just be tested on. It's a big issue. And who should gain compensation and ownership for new cell line discoveries? That's kind of an issue coming back to with Hayflick, where you didn't know if you necessarily could have control and ownership over it. And if the government just takes full control, whether you're private or government funded. And what are some impacts on the public health policy? So to begin, obviously there's enabled mass production of vaccines, and vaccine schedules became standardized by the 1990s, and I guess I said 1980s was when mass production began. Strict informed consent standards were implemented for fetal tissue. So there's the tissue transplant panel of 1988, where informed consent was certain. You have to know that that would be happening if your fetus was going to get tested on. You have to give consent, obviously. And then there's an NIH Revitalization Act of 1993. This was a whole big act, but one part of it was that a woman's decision in an abortion cannot at all have to do with the possible testing, so she can't have any knowledge that it could be tested on until after, maybe. So yeah, your decision cannot be affected by that fact before you make the abortion, just to slow any corruption. Scientists must now register human cells in regulated biorespiratories, and scientists must certify the identification of a cell line just to make sure there's no corruption and weird stuff going on. That's what I said, I swear. And Hayflick's settlement helped shape some U.S. policy by allowing some researchers to have more ownership of newly discovered new discoveries, whether it's federally funded or not, which is pretty nice. It's my personal reflection. I think the wi38 cell line has obviously affected billions of lives by being a vital tool in forming vaccines. I personally have gotten multiple, if not all of these vaccines. I'm sure I've gotten the majority, I hope so, as long as they're safe. I know me and almost every American is safe from terrible illnesses as long as they are not anti-vax. It is super cool how this one singular cell line from one randomly aborted fetus was actually saved millions of lives. I mean, that woman had no idea that how much of a big deal she had. I mean, we might not even be as technologically advanced as we are. I mean, you never know. We could have killed those deaths. The verdict could have killed someone, and it could have been something very important. We never know what our work could be like. I'm also happy to see researchers getting more credit in cell line discoveries and discoveries in general. It looks like Hayflick kind of helped with scientists having more control over what they can what they discover, their ownership over it. And now I get to learn about this. What are my personal thoughts on the ethic considerations? I personally do agree that there should definitely have been some sort of informed consent with the mother, compensation for those whose fetal tissue was tested on before without knowing, and possibly even now if they allow it. I think that the new policies are pretty good to make sure there's no sort of corruption and that women are not just having aborted fetuses so that people could just test random stuff on it, and I definitely think it's not necessary to have informed consent. I think I said that twice. Government also should not impede on researchers' own discoveries and new discoveries as well. On researchers' ownership of new discoveries as well as profiting off someone else's discoveries when they necessarily didn't want to, or they never said anything about trying to make a profit, or just control in general over it, over the monetization and selling of it. Yeah, I think Hayflick was right in that he should have some sort of say in what happens with the cells that he discovered himself, instead of the Institute and NIH and all that taking a lot of control over it, selling it, when he was just giving it out for free originally. And here is my works cited. All right, thank you. Have a good day.